Welcome back to Bioinformatics Introduction and Methods from Peking University. I'm Liping Wei from the Center for Bioinformatics at Peking University. We are delighted today to have Dr. Michael Waterman with us here today. We also have a few students in the audience who are here to speak with us. This is Dr. Waterman is a young boy, handsome from a very young age, as always. This is a historical photo of Dr. Waterman with Dr. Temple Smith, taken by Dr. David Lichtman. The main purpose for us to be here today is really to hear from Dr. Waterman himself how he and Temple Smith created the historically important Smith-Waterman algorithm. And he'll tell us there's a great story behind this photo. This is Dr. Waterman and Smith later at Boston University, I believe. In addition to his exceptional research career, Dr. Waterman also has done a lot of teaching and a lot of interaction with students, and he'll tell us the story behind this as well. As a pioneer and leader of the field of bioinformatics, he is also one of the three founders of the RECOM conference. Without further ado, let's welcome Dr. Michael Waterman.
Well, thank you very much for having me here on this beautiful campus, this amazing undertaking you're starting off in. I think you asked me to say something about my days as a student and how I got into this. I was from a rural place, and I majored in mathematics as an undergraduate because the courses at the beginning were easy for me. And they gave me a lot of room to take other science courses and lots of courses in literature and philosophy. But when I was a junior, I took a graduate course in mathematics, and I quickly learned that all math courses were not easy. Did my PhD on a field that's called dynamical systems now. Very theoretical PhD, but we use computers quite a bit. And I'd never taken a course in biology, and I had no intention of doing any research in biology. But at my first faculty job, I occasionally spent summers at Los Alamos National Laboratory. There was a great mathematician, Stanislaw Ulam, there. And he, for some reason, had the idea that there was going to be mathematics in this new biology. He didn't really know what it was.
When was that?
This was around 1971. And I think 72 or 73, I spent a summer there working on what turned out to be sequence alignment. And the goal was to align protein sequences and build evolutionary trees, which people still do today. And we had a very large data set. We had 27 cytochrome C sequences. And it sounds tiny today, but it was not tiny at that time. And we studied distance methods for aligning sequences and published a couple of papers. And later in the 70s, the protein data bank, Margaret Dayhoff, used the Needleman and Wunsch algorithm for aligning sequences, which was a similarity-based alignment system, a very strange algorithm. It did multiple gaps, but they were all weighted the same thing. So it could easily have been a quadratic algorithm, but their implementation was key. And as mathematicians, we thought that distance was obviously superior. and the woman who ran the protein data bank said, ligands. So we sat down to show that our methods were better. And the result of that was our paper, which showed that distance and similarity were two sides of the same thing. For the similarity algorithm, you could write an equivalent distance algorithm if the sequence alignments would do the same. And this quite surprised me, I must admit. So we had this in the back of our minds. In 1977, introns were discovered, where later a Nobel Prize was given for this discovery, and it was shocking. Perhaps today you aren't shocked by introns, but if all you know about biology is genes in E. coli, the fact that there were introns, that this stuff got snipped out and thrown away, it was just mind-boggling. And immediately we realized that the alignment methods we were using wouldn't work if you had to snip out pieces and throw them away. We didn't know what the problem was or what to do, but immediately we wanted to address this problem. And we failed for some time. A mathematician published a distance-based method for this. But if you think about it, 3 A's in a row are zero distance from 3 A's in a row. And also, if there are 3 million A's in a row, there's zero distance from 3 million A's in a row. So you had no way to distinguish between distances that were small. In 1980, we stumbled on to what's now called the Smith-Waterman algorithm. And the algorithm was there before the definition was there. Because if someone had given us the statement of the problem and that little paper we wrote, we would have solved it in 30 minutes. But we had no idea what the problem was. So these were very interesting times of stumbling around, trying to figure out what we should be working on. And it wasn't immediately accepted at all. I mean, it was. If you look at the citation buildup over the years, it's linear, and slow. It's very slow growth at the beginning, but eventually, I think people appreciated it. Perhaps because it's easy to code. It's also appreciated.
So in the first week of the course, I showed the paper, your paper in Journal of Molecular Biology. It's an elegant paper. We will teach the details in the course. It's very elegant modifications that did the trick.
Yeah. Yes. No. It's quite. It's subtle. It's quite subtle.
It's actually subtle, but it's simple elegance in the way that it was worked out. I think last time I checked in Google Scholar, it was cited around 8,000 times. One of the most highly cited papers in that science.
And hiding in that paper, when you read it, we give scoring parameters that haunted me for years. People would ask me about that. And I did the silly thing of making the expectation be 0. And then did something for gaps. And of course, I mean, if you take a fixed length sequence and you insert the letters at random and you make the expected score 0, that's not the same as the expected score when you allow that to vary all over. And I knew there was a problem there that eventually was calling Altshul's statistics, but it was a couple of years before we started working on that dimension. But that's always, when I see that paper, I remember the trouble it caused me. People would either believe we were promoting those as appropriate scores. Of course, no backing up.
So maybe you can tell us the story behind this photo. So where was it taken?
It was taken this is at Los Alamos, and just say the setting. David Lippman has an MD, and he did his residency in Arizona, in Tucson. His degree is from New York, And on his way, he stopped to see, he knew about Temple and I. He hadn't published anything in this area. He knew about Temple and I, and he stopped to see us at Los Alamos, and he took this photo. So I think it was before, my joke is, it's before he was David Lippman.
Right.
And some things about the picture, at Los Alamos you had to have a security clearance. And the security clearances were, go back, where I'll not touch this, were hanging off our shirt collars. That's how you did it at Los Alamos. Temple Smith was an Easterner. I grew up in the West, riding horses and so on. So Temple Smith was an Easterner, and so he's got the cowboy hat, and he's wearing a belt buckle that has a New Mexico in it. He's very proud of his cowboy gear. I had just spent a year in Hawaii, so that's why my Hawaiian shirt, and I think my belt is some sash that's hanging off. So, and this picture, David sent us this picture after he was back in New York, and I remember really liking it, and I lost it. And at some point, years later, I wrote to both of them and said, would you guys send me a copy of that picture? So, no one did. When I finally moved out of my office into a new building, this picture popped out of the work. So I scanned it, and it's, I think, viral at this point.
How did you meet and started to collaborate with Temple?
He was brought to Los Alamos by Stan Ulam.
What was his background?
His background was a physicist. He wrote a PhD in neutron transport and was getting out of that. And he had written at least one paper in biology that looked at the entropy of the genetic code.
Okay.
So he had done some work, and he and them got along, and then for years, when I was working at Los Alamos, Temple would come for a month or two a year, and I would work on these problems. I thought of it as my hobby.
How did it become a career?
Well, in the early, 1981, I decided to leave the lab, and I thought, what should I do next? And I thought I'd done some work in geology, and I thought about going into geology. And then I looked at the work I was doing in biology, and it looked like the most fun. And I thought there would even be data to check some of these things. I had no idea, of course. So I decided that. And I had went from Los Alamos as a visiting professor at the University of California, San Francisco. So that was when I became full-time, doing whatever this is.
And this next picture was taken at Boston University?
Yes, at Boston at Temple's retirement.
And this one?
And this one, I was for 5 years a residential faculty in a residence hall at USC. So I lived in the building where the students, I had a much better bedroom than they did, for sure. But I ate in the student halls, which where I think this was taken, went to events. We spent 20 hours a week doing one thing or another with undergraduates. And, almost none of them in science or engineering. It's great.
And you have to tell us about Recon.
Yes, so I believe this is 1997. And we wanted a conference that took statistics and computer science and biology all seriously. And the first conference was in Santa Fe. And Sony and Mistral really worked very hard to set this up. And it's usual at Recon conferences to have biology talks as well as science talks. But at this, and I started that actually at this conference, and the audience was shocked. They had no idea why a biologist would be talking about how they gather their data and so on. And today, when I go to the Recon conference, almost every attendee knows more biology than I do. The change in these years is amazing. Nobody asks why we're having technologists and biologists talking to.
the field had grown so much.
So, and really matured.
Yes, and you definitely helped pioneer it to get us where we are today. I also like to share with everybody that Dr. Waterman is receiving a friendship award from the People's Republic of China. It's a prestigious award in China for people who have made significant contributions to this country. So congratulations.
Thank you very much.
Maybe you could tell us a little bit about this as well.
You probably know as much about this award as I do. But I'd like to repeat a story I told you coming over here. When I was an undergraduate student, as I said, I took lots of literature courses. And I came on to English translations of Tang Dynasty poems. And I thought these poems were amazing. And they're amazingly modern. And they remain amazingly modern. The English translations, which leave all kinds of implications that are in the original poems out, but they have their own existence. But at that time, the only Chinese person I had come in contact with, I was at Oregon State University, which is an agriculture school. And in the house I was living in, once for a few weeks, there was a Chinese guy, but he knew no words of English, and I knew as many words of Chinese. And I just wondered about this country that was so behind a mist. I didn't know anything about it. So I'm so pleased that I've gotten to spend time in China and with the Chinese people.
You mentioned you came to Peking University campus in 97.
97, yes. And the streets were seas of bicycles with an occasional automobile. Mm-hmm. And of course, it's opposite today, yes?
Yeah. So maybe we'll give the students an opportunity to ask you some questions.
Sure.
If that's okay with you.
Of course.
Nice to meet you, Dr. Waterman. I'm Boxin Zhao from National Institute of Biological Sciences. I admire you very much for your great work.
As we all do.
Yeah. Today I have a general question addressed on teamwork. You know, many research milestones are made by a team, just like you and Dr. Smith, Waterman and Rouge, Watson and Crick. So, do you have any suggestions for us on helping a team function well? Thank you.
That's a very good question. In the small teams that I worked in, I think one of my rules is make sure the other people are really informed and really smarter than you are. So, and have different skills. And back at the time when I was working with Temple Smith, he knew much more biology than I did at that time. And he came from physics, so he had a physicist's way of looking at things, and I had a mathematician's way of looking at things. But now we have these huge teams where there are 50 authors, and that's a different order of beast, I think. So there needs to be, the boss needs to know what's really going on and bringing in, but once again, it's bringing in people with different skills who have a lot of integrity and are committed to the overall goal. Yes.
Well, nice to meet Dr. Waterman. My name is Wen Xiongzhou. In Peking University. My former major is biology, but now I'm working on engineering, and we are studying how to build a novel sequencer rather than simply the sequence. So I know a bit about the algorithm, especially in imaging processing. So I have a problem that is it possible to apply your famous algorithm to the problem of face recognition? So that we can know the figures in two different images are actually the same person, or vice versa, to apply current algorithms in face recognition to the problem of alignment, so that we can align not only DNA sequences but also protein structure, especially large complexes, or even gene regulatory levels. Thank you.
So, I mean, dynamic programming has a very great limitation when it comes to looking at comparing higher dimensional objects. And so we can write algorithms for two-dimensional matching, but they're not particularly useful. It really has its power in one-dimensional situations. And even there, of course, the computational complexity is high.
My name is Chen Xiaoyang from National Institute of Biological Sciences. And we know that there are many people transfer from other fields such as physics and mathematics or computer science to biology. So we feel very stressed. And my question is, in your view, what role does mathematics or computer science play in the development of bioinformatics, or in the future of the bioinformatics?
Yeah, I mean, I have to say it's essential. What else could I say? But having said that, it can't be just pure computer science or mathematics. It has to be integrated with biology, working on the right, with quotes, the right biological problem. And very often these problems won't have polynomial time algorithms necessarily, and you have to devise simulation methods or what's called machine learning. But I don't think bioinformatics can go on with that. If you made computer science and mathematics illegal to use in bioinformatics, it would die.
Nice meeting you, Dr. Waterman. I'm from Peking. Thanks for sharing us your experience. I have a question about, we know that doing research sometimes it is a very difficult thing, and when you face a really difficult time. My question is, did you meet some difficulty and want to give up, and how you come up with it?
My year on sabbatical at University of California, San Francisco, I got interested in the occurrence of non-overlapping patterns in sequences. And if you specified the pattern, one pattern going along the sequence is something called renewal theory. And William Feller worked this out after World War II. Beautiful chapter in his book about this. And I was thinking about restriction enzymes, and what about a collection of restriction enzymes? So you'd have a number of patterns, and they couldn't overlap with each other. So during that year, I started trying to work on this, and I would work on it a week and decide it was impossible. It couldn't possibly be done. And I would give up for a few weeks, and it would keep bothering me. I would come back and work on it some more. And eventually, now I can describe this in a 1-hour lecture, and it's understandable, and I just had to look at it the right way. But it took the longest time, and I gave up several times. It just crept around in my consciousness. It would just be there, and I would think, it can't be impossible, I would say, and tackle it again. So I couldn't leave it alone, is the answer.
I actually also have a couple of questions to ask you. With the next generation sequencing, the amount of data is increasing exponentially at an even greater rate. The sequence read archive, SRA database, is doubling every 5 months. From 2010 to 2013, the data increased by 100-fold, right? 100 times as much data now, as a 10 to the power of 15 nucleotides now, and it's going to double in another 5 months. So how do we keep up with this? No single library can keep such data. Even NIH in 2011 thought about discontinuing SRA because it's running out of budget. But fortunately, it changed its mind, so it's still supporting SRA. But as you see, it's even a problem for the NIH. How do labs like ours handle this?
I don't have a good answer for you, but we may come to the time when it would be easier, instead of trying to find something in the database, just to resequence that person again. And it may become so easy to sequence that keeping that reference material now may not be what I don't have a good answer for you. Everyone's struggling with this. In my building, at my university, in my campus, on the medical campus, and of course everywhere else.
It's a challenge, but it
really is, and it's not so easy to get this data from one place to another either.
Right.
Put it on a floppy disk and carry it across the room.
It also has opportunities for technical development, research.
Definitely. And serious people in computer science who study data compression and databases.
What do you think the future impact of next generation sequencing? It's already making great impact in biology. What do you think the future holds?
You've caught me, because often I'm asked to describe the future of bioinformatics, and I always answer by saying, well, tell me the future of biotechnology, and I'll tell you the future of bioinformatics. But you really ask about the technology future, and I'm just amazed at the sequence machines that have come out since the Human Genome Project. It's astonishing, and I think there's more to come.
And since you mentioned, what do you think about the future of bioinformatics?
Well, then I just no, I mean it follows the technology, right? And the biology, but biology follows the technology too. And some of these amazing single-cell imaging techniques to see different number of proteins moving in a cell. It's just that wouldn't have been interesting to think about in 1990, but it's on us.
Do you feel the field reaching a plateau or nowhere near it?
We're probably nowhere near it.
Certainly an exciting time.
And you think that there are incredible discoveries in biology that are almost as surprising as introns that are still happening. It's still a young field, even though I sometimes don't look at it that way, but in fact it is.
True. Wow. Well, thank you very much. This is really wonderful to have you here.
Thank you for having me.